All right, so this is a review for quiz 2, which is second half of lecture 2 and all of lecture 3. I'm just going to review some of the key concepts that you should pay attention to when you're studying for your quiz. Okay, so first let's go through the proteins. Right, so we have the proteins, right? And you should know that the proteins are made up of monomers. There are 20 amino acids. 20 amino acids. Okay, and some of them are essential, which means you have to obtain it through your diet. It's also a good idea to know the structure of an amino acid. I'm not going to ask you to draw it, but you should definitely recognize it and know the different parts. Right. So there is the central carbon right there. This over here, we call that the carboxyl group. Right, the acid group essentially. And over here, we have the amino group. Okay, so when you connect two amino acids together, the carboxyl group will connect with the amino group of another amino acid to form a peptide bond. Okay. Down here, we have an R group, okay, which is called a side chain. Right, and the side chain is different for everything, right? So like sometimes you might have like a CH3 here, sometimes you might have a ring of some sort here, right? Regardless of what this structure is, that's what distinguishes one amino acid from another, and we call that the side chain. Okay, so like on the quiz, I might circle a particular part of an amino acid and ask you, what are we looking at here? Okay, and you should familiarize yourself with these terms. Okay, so elements in a protein, right? We have the trio, right? The carbon, the hydrogen, the oxygen. We also have nitrogen, as you can see in the amino group, and trace of sulfur. Okay, so for the interest of time, I'm not going to ask questions in the quiz. I'm just going to, I'm not going to ask questions in the tutorial in this extra help session. Okay, I'm just going to be talking all by myself. That way we can cover more stuff, right? But if you have questions, please feel free to ask. So trace of sulfur. So some of the amino acids, they will have a bit of sulfur in it. Okay, and the functions, right? Functions, biological functions of proteins. So for example, they form enzymes, which help catalyze chemical reaction in the body, right? Okay. They can form protein, hormone, right, for signaling. Right? We learned about signaling in class today. And so protein hormones are made up of protein. There are of course lipid hormones as well, but that's a different story. And we have another function. Okay, proteins, they form important structures. Such as nails, right? Muscle, right? Hair, so on and so forth. So that's the function of proteins. I do want to talk a little bit more about enzymes. Okay. So enzymes, they work by lowering the activation energy, right? And in the lecture, you might remember there's a graph that looks like this. Right? This is energy on the y-axis, and then we just have time here on the x-axis. So your reactant is going to start with a certain level of energy, but you must provide extra bit of energy before the reaction is going to take place, right? And that extra bit of energy that you need to supply, that's called the activation energy. Okay, so it's the extra bit of energy that you must provide before the reaction is going to take place. So without an enzyme, the activation energy could be quite high. In the presence of an enzyme, you will be able to lower that activation energy. Okay, so that's the lowered activation energy. In other words, you will still be able to do the reaction, but you only have to supply a small amount of energy compared to before. So that's how an enzyme works, right? An enzyme works by lowering the activation energy. Now I'm going to give you a specific example of enzyme. I think we did something like that in class. Okay, so there could be an enzyme like that. Okay, and we'll call this like lactase or something. Okay, and there are these grooves where the reactants or substrate is going to be able to bind to, right? We call them the active site, right? Okay, so let's say you have your substrate here. Okay, the substrate. So this would be lactose, for example. And so lactase is the enzyme that breaks down lactose. And of course, the shape of the substrate has to be complementary to the shape of the active site. Otherwise, it's not going to be able to fit in, right? Okay, so the substrate will go into the active site, and the enzyme will carry out a reaction. And so in this case, the reaction would be to break the lactose back into its individual components, right? Okay, so afterwards, you will have the square plus the triangle here. And, you know, in our case of lactose, it will be a glucose and a galactose. Okay, so in this case, the substrate is the starting material, and the enzyme will lower the activation energy to carry out this reaction. Is that okay? All right, another big thing you are supposed to know for the protein is that it has 4 levels of structures, right? 4 level of structure. Okay, so I'll briefly summarize it for you here. We have the primary structure, okay, which is basically just the order, right? Order, the sequence of amino acids in the protein. Okay, and the bond that holds them together, right? The interaction that allows these amino acids to connect together, that is the peptide bond. Right? Remember earlier I was saying if you 2 amino acids, you can join the carboxyl group and the amino group together and form a peptide bond. Okay, so this is a type of covalent bond. Right, covalent bond. Next, we have this secondary structure. Secondary structure. So there are 2 things that exist in the secondary structure, right? So part of your polypeptide might form these coils, and then part of it might form these zigzag patterns, right? Okay, so they are alpha helix, right, and beta pleated sheets. Okay, so the amino acid is held together by the peptide bond, whereas the alpha helix and the beta pleated sheets, they are held together. They're supported by hydrogen bonds. Of the backbone, of the peptide backbone. Okay. Let me just pull a picture from the lecture notes to show you. Give me 1 second. Okay, so you can see in this picture, right? This yellow ribbon, that's the peptide backbone, right? Oh, it actually says right here, right? And that doesn't change, right? Throughout the entire polypeptide. What changes is the side chain, right? The R group. That's what's changing from amino acid to amino acid, but the entire polypeptide backbone, it's the same. Okay, and the interaction of the backbone, the hydrogen bonds that you form in the backbone, that's what's stabilizing the alpha helix and the beta pleated sheets. Okay, if you go one level higher, then we have the tertiary structure. Okay, which gives the overall 3D shape of the protein. Okay, and we have a name for that, right? What do we call the 3-dimensional shape of a protein? Does anybody remember that? What's a name for the 3-dimensional shape of a protein? If you're thinking about conformation, that would be correct. Okay, so this overall conformation, it's stabilized by interactions of the side chains, right? The R group, right? So some of them might be able to form more hydrogen bonds, some of them might be able to form hydrophobic interaction, and you don't have to memorize any of these interactions. They're just examples. The key thing to know is, if I ask you what stabilizes the 3D structure, you have to know is the interaction between the side chains. Okay, and finally there is the quaternary structure, right? Okay. Which are not always present, right? Only present in some proteins, right? And that's when you have multiple polypeptide coming together to form a complex. Okay. And what holds them together would be various interactions between the polypeptides. There could be more hydrogen bonds, there could be other similar forces, ionic forces, so on and so forth. Okay, so as we all know, right, I emphasize that in class, that the conformation is related to the protein's function. Okay, if you mess up the conformation, if you change the conformation, you're going to affect the function of the protein. It might become non-functional, it might be functioning the wrong way. And so, we try not to change the shape of the protein. But then in class, we talk about several factors that are going to affect the conformation, right? So factors affecting the shape of the protein, the conformation of the protein. Okay, and here they are, right? 4 factors we talked about. Specifically, there's pH, temperature, right? Type of solution that the protein is in, like is it hydrophobic, is it hydrophilic, right? That's going to affect how the protein is folded up. Salt concentration, right? So all these things, they are going to affect the shape, affect the conformation, therefore potentially affect the function of the protein. Now, it's also important to remember these things disrupt secondary, tertiary, and quaternary structure, but not primary, right? The covalent bond, it's too difficult to break by changing the pH and the temperature. Okay, but all these other things will be disrupted. Okay. Let me just double check if there's anything else you should know for the protein. I think you would be in a good spot if you start with those things and just focus on that for the proteins. Okay. Any question about proteins before I move on to nucleic acid? You can type your question. You can unmute and ask the question. Whatever you feel comfortable doing. Okay. Good, good, good. Next is nucleic acid. So of course we talk about DNA and we talk about RNA, and you probably should know the difference between the two. So a couple of things, right? So for DNA, we know that it is a double helix, whereas for RNA, it is single-stranded. Okay, double helix, double strand. Okay. DNA, it contains hereditary material. Right? The instruction. To make proteins. RNA, we didn't really talk much about the function, but we know there are different types. Okay, and we know some are copies of DNA, right? And in lecture 3, we learned about how some RNA is part of ribosome. Okay. So in terms of function, like DNA is just coding for the instruction to make the proteins. That's the material that gets passed on in when a cell divides. RNA, it's more versatile. Okay? Some of it's for copying DNA and transcription. Some of it makes up your ribosome. All right. We also have the type of nucleotide. Okay. So the DNA nucleotides, we have the phosphate group. We have a deoxyribose sugar, which basically it's a sugar that's missing an oxygen, plus the nitrogenous base. And there are 4 possible nitrogenous base for the DNA, right? There's the adenine, there's the cytosine, there's the guanine, and then there is the thymine. Okay. For the RNA, the building block is also nucleotide. Okay, so you also have the phosphate group. Instead of a deoxyribose, you will have the ribose sugar, not missing an oxygen, plus the nitrogenous base. Okay, so you have the same stuff basically. There's the ACG, but instead of thymine, you will have the uracil. Okay, which is U. Okay, so no thymine. Instead is replaced by uracil. What else do you have to know? I think that's a good comparison right there. Now, In terms of the bases, right? Like you remember we talked about it in class. There are two types. There is the purine, and then there is the pyrimidine, right? Okay, so you should know which one is which. Right? And I told you to remember pure as gold, right? That's the phrase. So A and G, that's purine, and these have two rings. Okay? Whereas the pyrimidines, it's the rest of it. There's the T, there's the C, and then uracil as well. And these are one ring, right? And you remember purines will pair with pyrimidine. Okay, so A pairs with T and C pairs with G, right? Okay. Now along that line, if you remember the base pairing rule, if I give you one strand, right, you will be able to predict what's on the other strand, right? So if A pairs with T and C pairs with G, you can just guess the other strand. So in this case, it will be T, A, G, C, A, G, G. And so this property, we say complementary. Okay. Now, what's holding the two strands together is basically hydrogen bonds. Okay. So there are two bonds holding the A and the T's, and three bonds holding the C and the G. Right. So C and G is stronger than A and T, right? Okay. So if you have two strands that had a high percentage of C and G, you will have a hard time pulling them apart compared to a strand that has a lot of A and T's, right? So that's what complementary means. Now there's another property that we talked about in class. That is the fact that they're anti-parallel. Okay, so anti-parallel means one strand goes in opposite direction as the other, right? Okay, and so, you know, if I ask you what are the two properties of a DNA double helix, right, make sure you know it's complementary and anti-parallel, and make sure you know what they mean, right? Complementary means the base pairing rule. If you know one, you can predict the other. Anti-parallel means one strand goes in the opposite direction as the other. Okay. So that is that for the DNA. Oh, actually, we did a little calculation in class as well, right? So we should do a sample calculation as well. Let's say there is 25%. Actually, 25 might not be a good number. Let's say we have like 10%, 10% cytosine. Okay. So how can we calculate the rest of it? Okay, so I'll give you a moment to try to do that. Okay, figure out the percentage of G, A, and T. Okay, and then we're going to take it up in a bit. Does anybody want to share the answer? What percentage of G do we have? For G, remember, C pairs with G, right? C pairs with G. So if you have 10% C, you would also have 10% G. Okay, because G, the percent of G always equals to the percent of C. All right. So we can add them up together. C plus G gives you 20%. Okay, and if you take 100%, subtract 20%, you would get 80% for the A's and T's, right? And we know A and T's are the same. So 80 divided by 2, you get 40% and 40%. All right. Make sure you know how to do something like that. Okay, I wouldn't be surprised if there is a question on that. I think that's pretty much it for the proteins and for the nucleic acid. Does anybody have any question before we move on to lecture 3? Okay, so let's do lecture 3. Lecture 3, you probably should familiarize yourself with labeling the cell. So let's take a moment and do that. All right, so here's your typical animal cell. And I'm just gonna label things as I see them, okay? So over here, you have the mitochondria. Okay, and I'm just gonna put like a brief description of what it does. Okay, so the mitochondria is the site of energy production. Yeah, that's where cellular respiration takes place, right? Okay, it's double layer membrane, right? Outer membrane, inner membrane, and all the structures associated with it. For something like a muscle cell, you would expect to have a lot of mitochondria because of the high energy demand, right? Okay, there's another mitochondria here. Over here, we have the nucleus. Okay, so the nucleus contains genetic material in the form of chromatin. Okay, it protects them, it houses them. And there's the nuclear envelope outside. Okay, there's a nuclear envelope. Now on the quiz, right, like I said, there's going to be 5 labeling. It's going to be the same diagram. And if I point to like this thing here, you can just tell me it's nucleus. Okay, like even if the pointer looks like I'm pointing at the nuclear envelope, just say it's nucleus. Okay, it's just the overall structure that I'm looking at. Okay, I'm just labeling these extra things to help you connect with the rest of the lecture. Okay, and then there's a thing in the center. Okay, if I specifically point to the center thing, then you have to tell me it is the nucleolus, right? And that, okay, I just have a private question. Nuclear envelope is same as nuclear membrane. Yes, same thing. Yeah. Nucleolus is site of ribosome production. Produces ribosome. Okay. And then they will exit, right, from the nuclear pores into the cytoplasm. Now this thing right here, we mentioned it at the end of class, so a lot of people might have missed that. That's the centriole. Centrioles. Okay. CE, centrioles. And you would have a pair of them in animal cells, but not plant cells. Okay. Their job is to produce spindle fibers. Okay. Which guides chromosome movement. Chromosome movement. Okay, during cell division, right? We're gonna pull the chromosomes from one side to another. You're gonna separate them, all that stuff. It's gonna be guided by the spindle fibers, and the centrioles is what's responsible for producing them. Okay, so again, Only found in animal cells, not in plant cells. Let's see what else we have. Okay, there is the rough ER here, right? You can see if you zoom in a little bit, I guess you can see all these blue dots on them, right? That's what makes them rough. So rough endoplasmic reticulum. Please spell it out on the quiz. Okay, don't just put like RER. You gotta spell it out for 4 marks. And the function of the rough ER is protein synthesis, right? Right. So works with ribosome to synthesize and package proteins into vesicles. Right? The vesicles will then bud off and then will fuse with the Golgi body here, right? Yeah, so that's the rough endoplasmic reticulum. Further out from the nucleus, we have the smooth endoplasmic reticulum. Okay. So in terms of the function for the smooth ER, it is the site of lipid synthesis. Okay, so think about the lipid hormone, right? The steroid hormones. Right, you will make that the smooth ER. Right, that's one function. And then the other function is to break down lipids. Okay, break down lipids. Detoxify drugs. Okay, and again, the difference between the rough and the smooth, not just the function, but the appearance is going to be different as well, right? The rough ER are the one that has the ribosomes on them, and the smooth ERs are the one that does not, right? And both of them are part of the endomembrane system. We have another thing right here that is a Golgi body, right? Or Golgi apparatus. Just stick with body. It's probably easier to write. So these are stacks of membranes close to each other, and they will receive the vesicles that bud off from the rough ER. And then what it will do is it will sort, modify, and repackage vesicles, right? From rough ER. Okay. And then that vesicle can go to different places, right? We wrote this down in class, but I'm just gonna verbally tell you, right? You should revisit that slide if you don't fully remember. So that vesicle, after it comes off from the Golgi, it could stay in the cytoplasm until you need the protein, okay? Or you can release it right away if you need it immediately. It could become part of the membrane protein, right? Embed itself in the protein. And you know, for my morning class today, we talk about various functions of the membrane proteins, right? So all those proteins came from one of these vesicles in the Golgi. And number 4, it might become another specialized organelle, right? Which is what this green thing is. Okay, so that is the lysosome, right? Lysosome. Okay. So various functions for the lysosome. Number 1, it contains digestive enzymes. for breaking down macromolecules. Okay. In class, we talk about diseases that would happen if the digestive enzymes were missing or not functioning. Don't worry about the diseases. I won't test you on that. What's another function of lysosome? Lysosome can also break down old organelles, right? Old organelles, right? It might play a role, plays a role in apoptosis, right? Which is programmed cell death. Okay, when a cell gets too old, you need to get rid of it for the benefit of the entire organism. And so the lysosome, it's going to release all its content, basically kill off the cell. We call that apoptosis. All right. I think those are the key stuff. I don't think I missed anything here. Any question about the cell? Okay. I'm just going to label the plant cell as well, because half of you will get the animal cell on the quiz. The other half will get the plant cell. So it's only fair that I also label the other one. Of course, you can find a fully labeled diagram in the study guide solution as well, right? All right. So just the obvious stuff again, that's the nucleus. All right. We have some new things that weren't there before in animal cells, such as this green thing. That's a chloroplast. Okay, so chloroplast is the site of photosynthesis, right? Contains chlorophyll, right? They're not found in animal cells. They are found in plant cells, and sometimes other cells as well. That's neither animal or plant. Okay, it's possible for them to exist in protists, right? That's what the amoebas and the paramecium are. We have another thing here, right? We have the cell wall. Right? Cell wall is made of cellulose. Protects the cell from bursting. Right here, that's the rough ER. Okay, we already talked about the function, like protein synthesis, worked with the ribosome. We have the smooth ER here. Okay, lipid synthesis, breakdown of lipids, detoxification, right? Like you might think, well, why would there be drugs in a plant, right? Well, if there's herbicide or something, then the plants need to process them. That probably happens in a smooth ER as well. Okay. We have the mitochondria here, right? Energy production. Right? There is this thing right here, this big structure in the center of the cell. That is the central vacuole, right? And the central vacuole basically stores water plus nutrients. In some plants, it stores pigment or poison, if we're talking about poisonous plant, right? But mostly water and nutrients. There is the Golgi body again. Right? Sorts, modifies, right? Yeah, that is basically that. Any question? Okay, good. Let me see if there's anything else you should know. There's a chart that talks about what's found in animal cells and what's found in plant cells, right? Maybe we should quickly review that. So, animal versus plant cells, right? So animal cells, there are lysosome, centrioles, okay, flagellum. Plant cells, we have chloroplast, we have central vacuole, and we have cell wall. Okay. Yeah, that's pretty much it. I'm looking at the quiz right now, in case you're wondering why I'm looking down. Yeah, that's a good starting point. Okay, so all these things that we reviewed right now, that's where you should begin your study. And if you know all these things in details, you probably will do quite okay, right? And then if you have time, you will review everything else for second half of 2 and all of number 3. Okay, I think that's it for this review session. Before you go, does anybody have any questions about anything that I did not cover here? Anything at all before we dismiss? I will post this note online, of course, and the recording. Okay, well, great. Thanks for coming. If you're in my afternoon class, I'll see you in an hour or so. Okay, have a good day, guys. Bye.